ideas. What you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior. And before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Hope I got that pronunciation okay. Thank you for joining me. And can you tell everyone a little bit about yourself and what you study?
Yeah, sure. Thanks for having me. I am a group leader at the Janelia Research Campus. What that means is that I have a lab here. I've been here for 8 years now. I'm not at a university. I'm at a research institute. We are funded through a charity. That means that I don't have some of the responsibilities that come with being a professor at a university, so I can spend most of my time doing research. And that's what we've been doing. And I think we're here to talk about some of this research.
Yeah. And so what is your field of study? Would you say that you're a computational neuroscientist? What is your field that you're in now, but also what's your background in terms of how you were trained?
Yeah, I would say right now I am a neuroscientist, period. I did certainly arrive here via computational neuroscience. It's not unusual for people in this field of neuroscience to need to have multiple kind of backgrounds, and they get here from all sorts of disciplines. In my case, I started with kind of a pure math undergraduate degree and kind of transitioning slowly into more applied math. Then the applied math became computational neuroscience. Then the computational neuroscience mixed a little bit with machine learning in my PhD that I did at the Gatsby unit in London. And then eventually my postdoc, I realized I can make more progress if I also do some of my own experiments. So that's when I actually picked up some experimental skills as well, started doing experiments. And that just kept going in my lab, where I now have kind of a full complement of kind of experimental side to the lab and computational side to the lab, and everything else that's needed to run such a lab.
Mm-hmm, mm-hmm. And so what does your lab study at a high level? What are the kinds of neuroscience questions and the kinds of systems that you're working with?
I would say at the very highest level, maybe, the lab studies just how network computations happen in the brain through kind of coordinated activity between all these neurons that we have in our heads, right? A lot of the complexity emerges from just the vastness of the neural networks that each of us has in their heads, but also that the animals that we study have in their heads. Something as small as a mouse, which is the organism that we study, has more than 1 million neurons. And if you put that in perspective, that's actually more than some of the biggest neural networks that we have today, the artificial neural networks that we have today. And so a lot of the complexity arises from the kind of high dimensional nature of this system. And I would say that where our computational kind of skills come together with the experiments that we can do is to really develop new methods to study such high dimensional systems.
Mm-hmm, mm-hmm. Before we get into like some of your work concretely, some of the recent research, some of the tools and the methods and all that, let's just talk at a high level about computational neuroscience and neuroscience more generally. You said something earlier about network activity. Can you define for people who aren't familiar with neuroscience or not intimately familiar, how do we think about things at different scales when we start to think about how neurons compute things and how information is encoded, going from individual neurons to circuits to networks? What do these things mean?
Yeah, good. Um, we can certainly start at a single neuron level, but one could start even below that, right? So like you said, there's multiple scales at which computation takes place. They all happen simultaneously, kind of all at once. And when we think of any one particular computation that eventually leads to something measurable, like I can reach my hand out to pick a cup, there was the effect of all these different scales and all these systems kind of computing, kind of propagating neural activity until it led to me reaching out my hand. And so it starts at the level of single neurons, which are themselves computational devices. Single neurons are very small, right? At something like tens of microns across, like a single cell. However, they have these processes that extend pretty far away relative to their sizes, their dendrites, their axons. Those integrate all sorts of inputs from other neurons. This happens at very, very small microscopic levels. Synapses get triggered. Voltages inside of this neurons kind of accumulate this activity. Ion channels open and close. So a lot of stuff goes on and on and on until kind of the output of this neuron, classically thought, is an action potential that travels down of its axon and then goes to kind of contact other neurons that are going to be receiving this message. And it's this property that makes it a network of neurons. Every neuron is connected to something like from 1,000 to 10,000 other neurons. And of course, they're all kind of recurrently connected to each other. Not every neuron is connected to every other neuron. So the exact properties of which neurons connect to each other leads to this kind of dynamic of exchanging messages back and forth with action potentials. This kind of dynamic through the particular structure of those connections leads to a computation and leads to this final outcome that is, me reaching out my hand to pick up a cup.
And yeah, I want to talk about the question of computation at multiple levels. We're just starting very basically. Like when we say computation, what exactly do we mean in biological systems? Whether we're talking about the single neuron level or the circuit level or the network level, most people have an intuition for what a computation is. You can have a handheld calculator in your hand or your smartphone and all of these computer devices that we've made. But when we think about biological systems, do we mean that they are literally computing things, or is this just kind of like a metaphor?
That's a good question. I think we can avoid, there's a whole, kind of debate about, is the brain a computer? Is it like a calculator? I don't think that's necessarily the right kind of debate. I think one way to think about it is actually not unique to the brain. It's the same for a lot of physical systems, that we make models of these systems. The models of these systems describe their behavior, to as good of a level as we understand it. And then we can make models of neurons as computational systems. Such a model would, for example, some of the simplest models are just a neuron as a perceptron. It's a great simplification, but we can imagine a single neuron gets 1,000 different inputs that arrive at different synapses with different weights. Those get kind of summed up, and the neuron outputs the weighted sum of those inputs. The model is clearly a computation. It's something I can write down and simulate in the computer. Whether the neuron behaves closely to this model is something that we now have to test, right, experimentally. We test experimentally. Such tests people have run before. In some cases, this can be a good approximation, can be a good model. In other cases, it turns out you have to model some more because, individual synapses might actually, for example, be on similar branches of the neuron, and they might have like a subunit of computation happening over there. And it's only the results of this subcomputation that gets kind of transmitted forward, and the neuron as a whole integrates all of that. So I would say the kinds of models that we make of neurons and of networks of neurons are very much kind of computing things. They have inputs and outputs. They transform a set of inputs to a set of outputs. Whether or not the neurons and the networks actually do that is on the experimental neuroscience to prove or to disprove.
Mm-hmm, mm-hmm. So maybe we can just at a very simple level, we can model neurons and networks. We can say, okay, we've created this toy model. It's a computer-inspired model. Some calculation is happening, like say something is being summed together. So if I think of like a handheld calculator, it's built electronically such that if a certain pattern is inputted, a certain output is reliably given. So if it's some sequence of digits and then the plus signal and then some sequence of digits, it will reliably add those things together and have a defined output. And at least some of the time, neurons tend to behave in that way. Just generally speaking, you can model them as something like a computational device.
Yeah, and like you said, right, a lot of the things that can get modeled that way, especially in a deterministic way, like a calculator would be, um, tend to be true on the smallest scale. Like if I raise the voltage of a neuron, I can predictably 100% make sure this neuron is going to fire an action potential, send that message down the axon. But then as it goes to kind of higher scales, now we have these networks of neurons that they're kind of coupled together strongly. Sometimes it doesn't matter which neurons fires the action potential as long as kind of the message gets relayed. And so you get into these less deterministic cases. Of course, physically, everything is still deterministic, but you get into cases where the behavior of a single neuron might be sometimes fail on individual cases. For example, me reaching out a hand, a neuron in my brain might activate 50% of the time that I do that. Another neuron might activate the other 50% of the time. So that's where kind of the network effects start to come into play, where it's not necessary for every neuron to behave like a calculator, just for the system as a whole is kind of at a larger scale to behave relatively deterministically. And then it comes to, of course, things that make us more human are the fact that we cannot always be reliably predicted, right? We do things that no one expects sometimes. And exactly how that kind of decision propagates from kind of the lowest levels of single neurons firing through to networks that, they may go one way or another in their kind of activation patterns, and me ending up saying one thing or another. That's the kind of fascinating thing that we hope that one day we'll get closer to understanding. And it's honestly one of the things that makes neuroscience pretty exciting to work in, because it's a way of understanding ourselves. And I think a lot of people get into it because of that.
What are some examples you might give of famous or well-characterized computations that happen in the brain, mostly at the single neuron level, just to get people started thinking about what's out there?
Some of the most famous models at the single neuron levels are the ones that led to Nobel Prizes. So things like the Hutchinson-Huxley model in their case, it was for the giant squid axon, which is one of the biggest neurons that you can find anywhere on the planet. That's good because it makes it easy to work with. It's kind of macroscopic. It's easy to record from. And what their model did, it was a model for the exact way in which an action potential might be triggered and propagate. Action potentials, they are the message. They're the central kind of unit of computation. So very important to understand exactly how these action potentials develop and propagate. And what made that research exciting from a computational point of view, that even back then, right, in the 50s and 60s, with pen and paper, they were able to make measurements, kind of extrapolate from those measurements a certain shape of a relation between ion channels opening as a function of time, and the voltage it generated in the neuron. And then from there, they were able to abstract away a computational model with a few subunits of computations in terms of different ion channels opening for different ions coming in and out, like calcium and sodium and so on. And then make predictions with that model that, kind of fit the data pretty well. And that was, frankly, one of the first kind of computational neuroscience applications. In some ways, it was the way that the field originated. Beyond that, in terms of single neuron computations, let's see what came after. Hubel and Wiesel are another famous example. They were recording from individual neurons in the brains of awake monkeys, while these monkeys were kind of looking ahead at a computer screen. And on that computer screen, various visual stimuli were displayed. And this was the first time when they were able to figure out what neurons in the kind of central brain, kind of higher up in cortex, what those neurons were responding to. So what were the messages about? The action potentials that these neurons were generating were recorded with an electrode, and the researchers were able to associate those messages to the exact stimulus being shown to the animal. And again, that was also done at a single cell level, kind of recording neurons one at a time with an electrode.
Mm-hmm, mm-hmm. And earlier, you said something, implicit in what you said was that this is a mistake that a lot of people make, especially early in their education, is that computations don't start at the neural level. They actually start lower than that. This is not necessarily going to be the focus of our discussion, but can you give people just a basic sense for computations that happen within cells?
Yeah, let me think of a good example of that. So a lot of neurons, especially the ones in layered structures, such as cortex or hippocampus, that means it's got multiple layers. There will be neurons that have a soma kind of in the deep layers, and they'll have an apical dendrite that extends all throughout these layers. And that makes the neuron kind of, the soma is the place where we usually look at for computations, because that's where the action potential originates. But then there's the whole kind of shape of this neuron with all its dendrites and the exact way in which these extend out. That is a computational device as well. You can get connections onto this neuron that are formed, let's say, in layer 1 at the very top. These tend to be kind of more intracortical connections from other neurons that are kind of traveling parallel to these layers. And you can have connections much closer to the cell body, where the cell body is in layers, let's say 4 and 5, where those connections might be from neurons, let's say in the thalamus, that bring in sensory information to this neuron. And then you might have kind of connections somewhere in the middle that are from inhibitory neurons in this network, which, for example, can modulate or like shunt the activity that's propagating either from the top to the bottom or from the bottom back to the top. And the fact that the activity, for example, the inputs at the top, they're so kind of far away, it means that for them to actually have an effect on the soma, they kind of need to accumulate or like to potentiate some inputs that happen more locally. And that kind of computation requires a nonlinearity. And the fact that it requires a nonlinearity makes this model of a neuron as a kind of a simple linear addition insufficient. It means you now have to think of kind of separately as the inputs to this neuron as being its dendrites, and then you can model the dendrites as different compartments. You can go into a lot of detail with this, and people do. You can model it with hundreds of compartments, where every compartment might in fact be the unit of computation. But again, that's not entirely known how many compartments you'd need to model a neuron very well. Single neurons have been shown to do nonlinear computations, such as the XOR problem. That's a famous nonlinear one that a perceptron cannot do. Turns out a neuron can do that.
So basically, every neuron, one neuron is one cell with one nucleus, and at any given instantaneous moment, it can send itself out only one action potential at a time, but they tend to have very elaborate physical structures, elaborate morphologies. So one neuron might send branches to different parts of the cortex. They might send them to different parts of the brain entirely that are very, very far apart compared to the size of the nucleus of the cell. And all of these different branches can basically take in separate inputs, have different sensitivities, different levels of
Yep.
information weight that they contribute to the whole cell itself. And so there's all these like little sub computations happening in different physical parts of the cell that one way or another need to get integrated before the computation at the level of that neuron kind of happens.
Absolutely, yes. And a good way to think about it is that the output tends to be fairly homogeneous, goes down the axon. It's the action potential. It regenerates down the axon. Even when the axon branches out, it goes to kind of all places, then it's pretty similar. The input is where different parts of the cell can behave differently. Mm-hmm. On the output, it tends to behave similarly.
Mm-hmm. So even at the level of one neuron, thinking about one neuron at a time in the brain, it's unfortunately not as simple as a simple calculator doing simple additions. There's a lot of complexity happening within that one cell before we even sort of scale this up to thinking about large populations of neurons.
Yes, that will be correct. The exact level to which that complexity comes into play is something we don't know yet, but we know that it's not as simple as a single unit of computation.
Mm-hmm. And so as we scale up our thinking to the level of the whole brain, ultimately in the organism, but just populations, large numbers of neurons in the brain and some chunk of the brain or something, what starts to change or become different in terms of what we have to think about and how the systems level effects and the dynamics are just going to start to behave differently than the single neuron level?
Yeah. So there's a few different levels before we get we get there, right? So we can kind of build up. So if we start from a single neuron, it might be embedded in a local network. This local network could be made, let's say it's a neuron in cortex. This local network will comprise kind of a small area around this neuron going up and down what we'll call a cortical column. A cortical column basically goes down through the layers of cortex, layer 1 through layer 6. Maybe, we can define it spatially in its 2D extent. We can define it as maybe 1 by 1 millimeters or a little bit below that. It tends to be a region of space where neurons are kind of highly interconnected to each other. And this pattern of connectivity tends to be repeated throughout, let's say, the entire cortex with a few modifications between different brain areas. It said this is basic. As soon as we put a neuron into this local context, which is still local, we're still not talking about brain-wide computation. It's still very local. The network dynamics complexity starts to come into effect. And that is given by the fact that, let's see, if I have two neurons, I can only connect them in a few different ways. Right? I can have one neuron excite the other one, and the other one could excite back or not. If I have three neurons, I might have a few more patterns of, which ones excite each other. As we start thinking of the actual number of neurons just in these local networks, which is on the order of 100,000 or something like that, probably even more.
And you're speaking about the rodent brain?
At this point, as far as we can tell, cortical networks have this property across at least mammalian organisms, right? Which have cortex. The kind of very high interconnectivity at the local level is the case across mammalian species.
I see. So even though the size of the brain varies considerably, when you get down to certain scales, the brains of different species can start to look at least approximately like one another in terms of their density and their overall connectivity patterns, even though at a different scale, things start to look very different.
Absolutely. And you'd have a difficult time distinguishing them if you got two pieces of different brains. The main way to distinguish them would be by thickness. If you got something that's very thick, like several millimeters, then you'd know that it's several centimeters, then you know that it's a primate brain or maybe a human brain. In a mouse brain, this thickness goes up to about 1 millimeter. As far as the density of connections, though, that is quite similar in the sense that in local circuits, there is very high connectivity. This leads to this combinatorial complexity problem that I was trying to kind of illustrate by going at more and more neurons, right? The more neurons you have, the more combinatorially many patterns of connectivity you can have between these neurons. And if we don't know these patterns of connectivity, which we almost never do, it becomes very difficult to even predict how a network of these neurons will respond to its inputs. Because while we may be able to control the inputs, let's say if there's like sensory inputs, just show an image, show a movie to an animal, we cannot really control what happens as soon as one neuron starts to excite another, and that excites another, and excites another, and you start having all this reverberation of activity in the circuit. It can lead to outcomes that appear non-deterministic because we're getting close to something that people call chaotic behavior. We're getting close to this chaotic regime where just slight differences in initial conditions can quickly diverge and lead to kind of different outcomes in the computation.
And when we start thinking at larger scales about systems and multiple circuits, populations of neurons, how do you guys start to approach this in the lab experimentally? Where are we at today in terms of technology, tools, and measurements for how many neurons that can be recorded and what the population size actually is that can be studied in something like a mouse?
Yeah, so that's where my lab and others, we've been kind of putting our effort and energies into trying to record as many units of these systems. And it is at this kind of medium scale where everything is connected to everything, where we can start to record at this point from tens of thousands, almost 100,000 neurons at the same time. There are trade-offs that we make in order to be able to record activity at these kinds of scales. Firstly, this is a lot up until not too long ago, 10, 15, 20 years ago, it was still common to lower single electrodes into the brain, record neurons one at a time. That was very kind of an arduous task. It was like a whole PhD of 7, 8 years just to put the data set together of a few hundred neurons, which then was like exploited and analyzed, very in depth for many, many more years after that. Nowadays, we have several techniques that allow us to record neurons more efficiently. Maybe I can start with the one that is a more natural successor to like the single electrode recordings. And these are high channel density probes. They're essentially still electrodes. They're kind of these long probes, except that you don't just get one channel of recording on them. You can get up to a few thousand channels, in fact. One of the most well-known examples, that it's not the only one, but it's called the Neuropixels probe. It's been developed over the past 10 years. We're now up to kind of better versions. There's been Neuropixels 2. There's been Neuropixels Ultra. Just last week, the NHP, non-human primate version of this probe was published. And these probes have really allowed people to record not one neuron at a time, but now something like, let's say, 500 neurons at a time. And then there's a different set of techniques that arose from kind of a separate, more, physics-y field, which are based around imaging. So instead of lowering electrodes into the brain, instead we put a big microscope above an animal. We replace a piece of the skull of the animal with a piece of glass, and we put certain molecular proteins inside of the neurons that will fluoresce when the neuron activates. When the neuron fires an action potential, there's a lot of calcium coming into the cell. It binds to these proteins that most of the time we use what are known as GCAMs. These are genetically encoded calcium modulated proteins. When calcium comes in, because of the action potential, the cell is nicely fluorescent. And if it's fluorescent, that means you can see it with a microscope. And that's what we do in the lab. We have been kind of building better microscope and better kind of ways of recording from a large number of neurons with microscopy.
Mm-hmm.
And those are the techniques that get us to those 50,000 and 100,000 neurons.
Yeah. So when we talk about the types of animal research that you do that we'll come to, when we talk about recording from large populations of neurons, there's essentially two types of ways of doing that. One is to stick a whole set of tiny, tiny, thin little electrodes into the brain. These are like tiny little thin wires. Each wire can have multiple channels on it. And each of those channels can pick up a signal from one neuron that you can isolate. But because you have many of these electrodes on the little implant that you put in an animal's brain, you can record from hundreds or even thousands of neurons potentially at a time. So you've got action potentials that you can record from a bunch of different neurons in some little chunk of the brain. And then there's this other technique, which is a microscope-based technique that combines, it's basically like the things from physics coming together with things from molecular genetics, where the optical capabilities to point a microscope at a brain and record the image were developed together with techniques that allow you guys to engineer cells, neurons, to literally light up, and then you can see them, literally watch them with a microscope. And so these two techniques are now at a place, and they both have, pros and cons in different ways, but we've got two different potential ways to record from thousands or even tens of thousands of neurons at a time, meaning that we're talking about individual neurons. We're not talking about like an fMRI signal that smears everything together, correct?
Correct. Yeah. And let me tell you, that's making projects much faster. It's making our ability to reason about this processes, the computations and the behaviors much faster. We don't need to wait for, many years to collect a data set. We can go into the lab, collect the data set already with thousands of neurons, kind of analyze it, and then come back the next day and based on what we analyze, just kind of set up a new experiment and collect a new data set. Mm-hmm. It's really been empowering, a lot more people to do these kinds of experiments too. Yeah. And one of those is myself, because I have a computational background. When I was, doing my PhD, I never thought that I would work in a wet lab at all, until, it was made sufficiently easy and approachable that even, someone with a computational background like myself imagined themselves doing it. And then it turns out, it's not as difficult as we thought.
One thing I want to touch on before we get into some other stuff is when you start to think about it, if you're recording from 1,000 neurons or 10,000 neurons or whatever, the brain is ultimately an organ with millions of neurons, many, many millions if it's a primate or a human brain. But even a mouse brain has millions of neurons, as you said. This is more, I think you said it's more than the artificial neural networks that are doing all of the impressive things that we see in the AI world. Yep. So even a simple animal like a mouse that's nowhere nearly as sophisticated as a human primate is already this very high dimensional creature with many, many neurons. When you're recording from that many neurons, then we remember that neurons are expensive from a metabolic perspective. Even just one action potential, think about all of the protein channels that have to open and close, all the parts that have to move, all of this stuff takes energy. When we, I'm trying to anticipate some of the more computational questions we'll get to, but when you're recording from this many neurons and starting to think about how the brain encodes information, how do you have to anchor that in thinking about metabolism and efficiency from like literally an ATP perspective?
Yeah, no, that's definitely a good approach to come at this problem. And it turns out it's one of what I would call kind of several lines of computational thinking that has moved us over the years. And this goes back to maybe as far back as the 90s or something like that, when a lot of kind of what we think of as the biggest computational theories arose. What you're describing, or the way that people thought to put that into a model is to think along the lines of how can I achieve a given computation? So I can write down a computation. I want to achieve this with the smallest number of spikes. So by minimizing the total number of spikes across the network that I'm going to generate in order to produce this computation. Then turns out that is a constraint that we put on a problem. And those kinds of constraints can kind of limit the space of possible solutions to the problem. And it's one line of modeling to create models of this sort that have these kinds of constraints, efficiency constraints, and then see how well they match things that we see in the brain. This has taken a lot of different forms. I'm just, the efficient coding hypothesis is kind of its main kind of umbrella. It can apply pretty well, and there's been some good predictions made at the level of, for example, sensors in our retina. Turns out, those operate in a pretty efficient way, and they tend to align with the predictions of efficient coding theory. They can also align, the model can work also pretty well in species that are pretty small, something like a fly or, in that region, an insect, right? And an animal that still has to do pretty complicated things like, fly around the world without bumping into things and avoiding predators and all sorts of things like that. But it has like now it has a very limited kind of computational budget because it has relatively small number, it's still like 100,000, small number of neurons, a very limited budget energy-wise. There, the models can still apply pretty well. Now you come to something like the human brain,
Mm-hmm.
and all of a sudden, those efficiency constraints start to kind of not apply as much anymore.
Meaning the predictions of those types of models don't do as well.
They don't do as well, and they can only go so far.
Mm-hmm.
Because it turns out that this is a constraint. Efficiency is a constraint. You cannot let the constraint limit the kinds of things you might do. What I mean by that is bigger brains, especially in humans, they have to solve these kinds of complicated problems that we are not even sure how you would solve them at the level of a neural network. And so kind of driving that by a constraint that you put on the brain isn't a good way of finding out what the problems are, or like finding out what does this network compute. Yes, maybe, it's not going to go way out of its budget of ATP in order to do that. But at the same time, if we're looking at all the action potentials that are happening in the brain, we're looking at a lot of the kind of action potentials that happen, even, for example, in something like a very sensory area when our eyes are closed, for example. We do this in mice. We record neural activity in complete darkness in their visual cortex. Turns out there's still a lot of activity. It's structured in certain ways. We understand some of it. But it's certainly not completely quiet. As would be the prediction of efficiency as a fundamental principle.
Mm-hmm.
Yeah. And like my perspective, and it's kind of we could explore this in different ways, but I would say my personal perspective on it is that brains and networks that are this big, they find certain solutions to these problems that are kind of kind of idiosyncratic. They're ways of connecting neurons that you wouldn't have thought about, that maybe like happened by chance at some point during evolution. The places that are kind of hard to escape from and really kind of optimize efficiency, which is why the brain is not very efficient, right? It consumes 10 times more energy per cell than any other part of the body.
So, I mean, it's really easy to get really into the weeds here. We have to keep in mind some things simultaneously for probably the rest of the discussion, which is nobody knows exactly how this stuff works. There's many different things that we can talk about and many different perspectives we can share. So we'll try and do a good job of explaining, there's this model or this theory, and then there's this other way of thinking about things. But one thing I want people listening to keep in mind is two separate but related questions are: how does the brain compute the stuff that it computes or that it's doing? But then there's also the question of what is it actually doing? Like
Yeah.
before, and you kind of need to think about both separately and together in parallel to start to think about some of this stuff. So I'm going to ask you a big question here, and there's no answer. I'm just making that clear for everyone. You can give your perspective and talk about maybe some of the other perspectives that are out there. But I just want to ask you the general question. What do you think, and what do neuroscientists generally think the brain is actually ultimately built to learn and encode? So, for example, is it built to learn and encode sequences of actions and to figure out how to do that from experience as efficiently and effectively as possible? Is it built to construct representations of stuff outside the organism, or to abstract regularities that are these more abstract things? Is it all the above? Can you start to talk about maybe your perspective and some of the general perspectives on this?
Yeah. I'm sure it's all of the above. But perhaps I could share what I think might be like one of the more crucial ones. And I think one of the more crucial ones for humans, and we'll see when we get to study this in animals, there's certainly people going that way, is that our brains are very good at making models of the world, kind of like you said, like representations of things outside, making models of the world. But also they're very good at running those models forward in time. Right? In our heads, we can imagine what would happen if I did this or if I did that.
Yeah, mental time travel.
That's right, but into the future. And that is an extremely important skill to have, because if you can predict how the world is going to react to your actions, you can avoid some of the most costly actions that you could take.
Yeah.
And you could really kind of find or narrow in on what are the good actions that you can take. And I think a lot of that depends on not only on having a model inside of our head of how the world works, but also our ability to kind of it's generative AI, right? But in our heads, for kind of extrapolating into the future, how the world would change, if we do this or that, or if we don't do anything at all, which is also very much something we do all the time. Why do I think that's one of the most important things? It's fairly, we think it's more unique to us humans. We build on top of that ability very much. I think a lot of the knowledge that we transmit abstractly between people, and it's knowledge about how things work. It's knowledge about models of the world. We make models of the world and we transmit it to our children. We transmit them to other, members of our tribe. We transmit them through society to, our whole thing is to make models and to transmit them forward.
And, all of these things, you might emphasize them or de-emphasize them based on the person you're talking to might have different opinions on which ones to emphasize, but also these things might exist to different degrees in different types of organisms. Like obviously a centipede is presumably not making the types of abstract models that a human being has, even though both animals need to, for example, chain together sequences of muscular motor action patterns in an efficient manner. So when we think about mice, how do you start to think about this in mice, and what the type of brain a mouse has needs to do? Are mice capable of abstraction a little bit, or like how do you start to think about what the mouse brain is built for?
Right. We think so. Let's see, what's a good place to start? My lab more directly works right now on how animals like mice make models of visual environments. So not necessarily how they, extrapolate models in their head. That's, I think, something a little more uniquely human, although we have seen it in other people's work, for example, in areas like the hippocampus, where we see replayed patterns of activity that correspond to the intention that an animal, like a rat or a mouse, has of where to go forward. And that's pretty exciting research because it's very similar to the kind of generative properties that we think are useful for simulating the environment.
Mm-hmm.
We work on the part of like how do we assimilate properties of the environment. A lot of that has to do with forming memories of one kind or another. In our case, because we're more of a visual systems lab, we look at how these visual memories are being formed. And it turns out there are certain parts of the visual system that develop representations of the world, develop these visual memories as an animal experiences it and becomes very familiar with it over time. We know they're new because they're not there, kind of on the first day of training. We don't have these very selective neurons that respond based on the visual environment that the mouse is in. These get developed over days. And we find that animals can use these representations to respond, well, to drive their behavior, first of all. So being visually familiar with certain environments allows them to afterwards quickly learn which one is going to provide a reward for them. Reward training, kind of reinforcement learning, is something that a lot of animal labs do in order to get animals to produce an outcome of a computation. But then we also find that these representations kind of develop by themselves as animals just kind of explore these worlds that we create for them, rather than them needing the animals to kind of forage for rewards. The foraging part isn't necessary in order to build an internal model of the world. And yes, we find neurons that seem to behave like that in the mouse brain. And so we're kind of, we just had a paper published about that, and we're continuing that work going forward.
Mm-hmm, mm-hmm. When we start to think about the population activity of neurons, how a large number of neurons behave and what these patterns do and how they encode information and stuff. So one thing to talk about maybe is the general rhythm or pattern that the population has. How does one neuron in the population behave with respect to other neurons in the population? This is where we start to talk about things like the correlations between neurons. And when you get into the weeds here, this has different implications for how information can be encoded by neurons. But trying to tie this to something that people might have some familiarity with, people might have an intuition that, for example, when you go under anesthesia or you go into non-REM sleep, you're unconscious. So there's sort of less going on in terms of conscious experience. The neurons in that state tend to have a pretty rhythmic and correlated pattern of activity. And you see something very different when an animal's awake, and especially when it's awake and it's like focused and thinking about stuff. Yeah. With respect to populations and the correlations between neurons, how do we start to think about the correlations they exhibit and how that relates to things like the robustness or efficiency of encoding and attention, and just how different types of information encoding might be allowed or disallowed with these different types of correlation patterns?
Yeah, let me start actually with the example that you gave, because I think it nicely brings in together two ideas we discussed, the sleeping or anesthesia example. Turns out, yes, it's oscillatory activity during sleep a majority of the time, except for REM sleep. But that's actually a lot of action potentials. Mm-hmm, it's not a fundamentally reduced state where the brain is trying to conserve energy or minimize action potentials. Mm-hmm. It's still more or less the same energy budget when we're sleeping and like technically not doing anything with our brains.
So right? It's just a patterning difference.
It is definitely a patterning difference. The exact role of sleep, as is not well understood. Whether it has something to do with kind of cleaning out your brain and clearing bad stuff or things of that sort. But we do know something very interesting during sleep, and that is that we dream. And dreams are kind of the fundamental generative models in our brains being played. forward. They very much depend on the experiences we're internalizing, right, over the course of the day. Sometimes over many, many years, we can dream of things that happened a very long time ago. So dreams are a great example of something that happens during sleep with patterns of activity that sometimes can look like the ones during wakefulness, sometimes they do not. Now, you pointed it out as an example of kind of network dynamics operating differently. And that's perhaps one of the, I would say, most clear differences that we can study and think about in terms of network dynamics, because it's so very clear that under anesthesia especially, and under certain forms of like very deep sleep, neurons are very much coordinated. They might fire a lot of action potentials in a burst, then quiet, then burst, then quiet. These are what people call up and down states. There are models of up and down states. We built a model when I was just kind of starting out in my neuroscience career. We built a model of that. There's other models of how up and down states might be produced. It can be a shift, for example, in the amount of inhibition that a network has to play with. We think that more inhibition kind of stabilized regime prevents some of this kind of runaway excitation that leads to these bursty population events. But ultimately, we think that that's just kind of a way of correlating a network to the point where it's not really able to do the things it's normally supposed to be doing. It's just a way of kind of impairing the normal neural computations, and perhaps that impairment is what leads to the loss of computation.
So at a very high level, the extent to which neurons are firing in a coordinated fashion will determine what they're computationally capable of. Clearly, the neurons, as they're firing the ways they do during anesthesia or deep sleep, they're clearly not capable of doing the things that they do when they're awake.
Yes, except for dreams.
Except for dreams.
And I was just saying that's kind of in some ways it's something that we call a one-dimensional pattern of activity. If all the neurons go up and down together, this is the whole population having one mode, of coordinated activity. We can project the neural activity onto a single vector direction in neural space and get most of what's going on, most of the activity going on over there.
But when these generative systems are in play during dreaming or during waking, that's not the case.
That's not the case. Then, this one-dimensional mode of like the whole population activating or not becomes very uninteresting, has very little activity in it. It really becomes this very high-dimensional pattern where there's all sorts of groups of neurons that might be activating together sometimes. And really, there's no good way, at least for these kind of cortical recordings that we do, there's no good way of reducing that to say, it's not one dimension, but maybe it's 5, maybe it's like 10 different axes, and that's all the brain is doing. No, we don't see that in any of the scenarios we've looked at. There is not a way to reduce activity of, in our case, 10,000, 100,000 neurons that we record. We have no way of reducing that to, let's say, just 5 dimensions.
So people have tried and failed to come up with models of the brain and to analyze these high dimensional data sets where you've got many neurons recorded. And people have tried to say, okay, what if we can explain everything? If we can explain the sleeping brain in 1 dimension, maybe we can explain the waking brain in 3 or 5 or 10. And we can't do it.
Yep, absolutely. And we can also, we can prove that we can't do it, right? As we record more and more neurons and we get more and more samples of this kind of neural data, we can develop the kinds of analyses of the, if you want, of the mathematical properties of this kind of cloud of points that the neural activity inhabits. We can quantify those mathematical properties. And those quantifications can tell us, for example, that, there is no linear, for example, set of dimensions that you can project into that span the whole space, but also that there's no kind of nonlinear set of dimensions. So nonlinear manifold, which is where it gets more complicated and more mathy. And then you have to think of kind of the local properties of this mathematical object that we call a manifold, because it resembles, the mathematical concept of a manifold. It's a kind of a curved space, but linearly. Locally, it's still linear. Locally, it still looks like a low-dimensional space. We don't see that in the brain as soon as we record enough neurons. If we record very few neurons, or if we put the brain into a very kind of low-dimensional state, for example, I don't know, ask an animal to do the exact same thing 1,000 times, which is what a lot of experiments are actually like.
Right.
Then, yes, it looks like there's very little going on. But as soon as we ask the animals to do lots of different things, or we show lots of different stimuli with different properties, we see that the brain kind of, the neural activity just jumps around in this space and just bounces into all sorts of different patterns.
So it's not just that the brain activity of a waking animal doing interesting things is many dimensional. You can actually prove that it's sort of this hyperdimensional thing that can't even in principle be projected down to a linear combination of, even dozens of dimensions.
That is correct. Yes.
And what do you think that means in very, very simple terms? Is that connected to what you said before, perhaps, that, this has to do with the idiosyncrasies of evolution and biology, and that, things aren't built from a blueprint by an engineer. They're kind of cobbled together by this random walk through evolutionary space over time.
Yeah, no, I think that's probably a part of it. It's good to think about it in terms of information coming in, let's say, to our eyes. We're studying a visual system, and what's going out. What's going out is actually the output of our system is fairly low dimensional. We can really describe what we do on a moment by moment basis with, just a few bits of information, right? However, the amount of information coming in through our very high dimensional sensors, our retinas are some of the best cameras on the planet, basically. This is extremely high dimensional information. As we walk around, we're bombarded with very high dimensional information, lots of photons in all sorts of configurations that our brains are somehow very well optimized to extract information from and distill it down to these very few bits of information that go out, which constitute our decisions, right? We see a familiar face, we say hi, that's the output. The computation itself has involved like a very high dimensional process. Now, what's really kind of surprising is even as the data coming into our retinas is so high dimensional, it appears that the way the brain solved this problem is to make it even more high dimensional inside of the brain. So we go from something like 1 million retinal ganglion cells. I might have my numbers wrong. I know the factor of expansion. I might not know the absolute numbers.
There's a lot.
There's a lot. But then the factor of expansion of how many inputs come in through the retina and through the ganglion cells and through our nerve, optic nerve, it expands by a factor of 50 or so inside of our visual cortex. So for every little single pixel, let's say, in our field of view, there's another 50 neurons kind of somehow encoding that pixel in different ways, let's say. But they're not encoding a single pixel, they're encoding patterns of pixels.
Yeah, there's more ways to encode the pixels than there are incoming pixels.
Yeah. And so that's right. Another way to say, like, dimensionality gets expanded before ultimately it has to collapse, because our actions ultimately are relatively low dimension.
In principle, in theory, what would be the possible advantages of doing that dimensionality expansion before going in the opposite direction? Yeah, in principle.
So, there is an empirical answer to this, which is that when we try to design artificial neural systems, right, artificial neural networks, this kind of expansionality is beneficial and these systems work better, the more neurons they have to work with.
So that's just, we know that's true from that perspective. So we know it's true even if we can't really explain why.
We have a few, actually, it turns out the theory for explaining why was developed before the neural networks were developed. And the neural networks were not developed necessarily with this theory in mind, but this theory goes back to kind of statistical learning, I believe is the name. No, I may be wrong about this. This is not, some of the initial ways of thinking about it is if you have a kind of a nonlinear computation you have to do, what you could do is take your inputs, project them into a high dimensional space, and then do a linear computational in this space. That's much easier to do now. There are actual mathematical proofs that this kind of thing works almost always. Some of those proofs are related to neural networks that say a neural network with an intermediate width that can go very large as infinite can do any computation. As opposed to a perceptron that cannot by itself. But when you put many of them, infinitely many, kind of in a row, it can do any computation. So those are the kinds of theories that talk about dimensionality expansion. In practice, with neural networks and artificial systems, we don't just make them high dimensional, we also make them kind of hierarchical and we make them sequential. So the input keeps getting processed and processed and processed and processed.
So basically the idea here, the simple idea here is that if you do this dimensionality expansion, it can make computations easier to perform, and then you can project them down into a lower dimensionality and then execute ultimately a motor action.
Correct, yes. That is the idea that has mathematical support. I would say artificial neural networks both validate that in the sense that they do expand the input into a higher dimensional space, but in some ways they also invalidate that because they also require kind of many repetitions of this computation to happen. You don't see that as much in the human brain. If you look at the human brain or any mammalian brain, some of the early visual cortical areas are very large. So we've got what's known as V1, the primary visual cortex, and V2. These alone take up, depends how you define it, but by some definitions, you could say in the human brain, they take up something like a third of all the visual processing areas. In something like the mouse brain, they take up more than half easily. So then there's a lot of these higher order computations are actually happening in smaller and smaller and smaller brain areas.
Right.
May also correspond to kind of the space contracting that way.
So let's start. Well, actually, let's go to one more place before we talk about some of your recent experimental results to set this up. So there's a notion, you see this both in the machine learning and AI world. So people may have heard this in some context, even if they're not researchers in this area themselves, but it's the notion of supervised versus unsupervised learning. Let's just start with a basic definition of those two things.
Yes. These are coming entirely from the machine learning field, where supervised learning is any computation that you set up where you have labels that you're trying to predict from inputs. So it's another way of saying a regression problem. People might be very familiar with linear regression. That's the most fundamental supervised learning problem. You've got inputs and outputs. You model them as a linear operation. But you can also predict the outputs from a deep neural network with as many parameters as you want. And that's also supervised learning because you're predicting labels, such as, in this image, there's a cat or there's a dog or, one of the many labels you could put on an image. And you have a big data set of such pairs of inputs and outputs, inputs and labels, that you train your classifier on. And that's typically described as supervised learning. And it's been the foundation for almost everything in machine learning these days. On the other hand,
So
there is
a supervised learning algorithm would be one where the algorithm is given the things to look for or to do ahead of time. It's given some information and kind of told what to do.
correct. Yeah, it's instructed on a kind of a case by case, an example by example, what to do. And if you have a big enough data set, then you will be able to hopefully know what to do in a new situation, a new input that you have never seen before, still able to produce the right output.
Mm-hmm.
On the other hand, unsupervised learning is something that has also been kind of a big idea, and it's only becoming more practical these days. But it's been a big idea for a while because you realize pretty quickly when it comes to human brains and animal brains that the amount of pure supervision that we have is very low. There's very few times that, a parent points to something, says, that's a dog, it's a zebra, we get some of this, but it's very low compared to what any of the artificial systems these days get in their training. And instead, it's been thought for quite a while that the brain must also be able to do some kind of unsupervised learning where it can extract patterns from the environment without necessarily being told what those patterns are. And the reason why it made sense to think about it that way is because our environments are in fact very highly structured. The photons that arrive on our retina are not just random combinations of things. They arrive in like very specific patterns. And being able to abstract away and kind of make certain, understand that certain combinations of photons, certain patterns are much more likely than others, that's a form of unsupervised learning. Typically, and especially from a pragmatic point of view, let's say you have an artificial system you want to do this in, it is very common these days to do something like unsupervised learning to prepare a system for the eventual supervised learning that you're going to do with potentially a much smaller set of labeled examples. And when you do it that way, we call the unsupervised learning as unsupervised pre-training. We're pre-training a system to abstract away the kinds of patterns it sees in the data so that when labels come later, it's able to more easily associate those labels with abstract concepts in the data rather than with the kind of pixel level properties, which might not be a very good association.
Mm-hmm. Mm-hmm. Okay, so there's a notion that unsupervised learning can help an organism learn the general, more abstract structure of stuff. And then when it has to do more concrete learning, like, matching pictures together or generating an action when it sees something, like in some of the experiments we might describe, that what you might call supervised learning or task-based learning, that can happen faster if there's already kind of some scaffold there that emerged in this unsupervised manner.
Yeah, absolutely. That would be the idea. And it's been a very popular idea with theoreticians for quite a while. There's quite a few algorithms or models, let's say, that have made predictions of what things would look like in the brain if unsupervised learning was going on. And a lot of those were promising. And so a lot of people in my generation, for example, kind of seeing those kinds of models, we've been very kind of inspired to like go and find more directly something of that sort happening in the real thing, right? In the real brain and with real neurons. And we think that's what we found.
Do you want to set up some of these experiments a little bit? What is the general experimental paradigm you're using? What does it look like? What do the mice have to do?
Yeah. So in this particular paradigm, mice are head fixed on an air floating ball or kind of a treadmill. Turns out mice are very good runners. Just for the same reason that hamsters have, kind of naturally run on hamster wheels. Just about all rodents, they just kind of like to run on wheels. So we had fixed them on wheels. We have them run. And then we pick up that signal that, the spinning of the wheel, and we couple it to a virtual reality setup, which consists of monitors arranged around the mouse. And as the mouse is kind of running and moving forward, it moves forward in the virtual reality, and it gives it an impression that it's traversing some environment.
Mm-hmm.
Now, that might seem abstract and matrix-like. The reason we do this is so we can use our fancy, sophisticated, very large microscopes, right, that are kind of fixed in place. That requires us to fix the animals in place as well. And then we can come close to with the microscope and record from these very large neural populations, while the animal is kind of having an experience that's, not exactly natural, but it has certain of the same features as a natural experience.
So the mouse is literally running on a ball, and it's looking at computer screens that have what people might think of as kind of like a video game-like environment, usually a maze of some kind. And
Correct.
The screen and the way it moves and presents pictures to the animal is tied to the animal's running its motion on this ball. And so it's very much as if the mouse is actually running through a physical space. And for all intents and purposes, it certainly looks like the mice think they are.
Yeah, we don't know what the mice think. And we don't think that we are, we give them a little more credit than that. We think they do know. However, we think they're processing things in kind of the same way, or like reusing some of the same computational computations in their brains to process this kind of data. I don't think they're imagining that they're not fixed in place anymore. For our purposes, this has a few beneficial properties. It's a departure from some of the more classical experiments. In classical experiments, stimuli are kind of shown statically, or they're kind of abstract stimuli shown on a screen in front of an animal. In our case, these stimuli are presented in a way that the animal might experience them if they were running around.
Yeah, it's a dynamic environment. The animal has to use its navigation abilities. Things are, it's the stimuli are tied to the animal's motion through this virtual space. So it's obviously this is an artificial setup, but it's quasi-naturalistic in that sense.
Yes, and we think it's part of the reason why it can drive the kinds of learning processes that would happen under naturalistic scenarios. And when we started this type of experiments, what we did is we set up kind of these different corridors, different regions of the world. We put different naturalistic pictures. Really, they're photographs that I took with my phone of like different visual textures around the world, really, not just here. And then we put these on the walls, and we wanted the mouse, to learn that some visual textures are rewarded and some are not. Some environments visually are good environments. Some environments are kind of boring. They don't have anything in them. We train the animals to respond to those good environments, the rewarding environments, to recognize them and respond. These animals are water restricted, so they're motivated to work for water rewards. And that is how we get them to report to us. Whenever they lick, it's a report that they think they're going to be getting water in this environment. And then after training for anywhere from 2 weeks to 3-4 weeks or so, the animals are reporting on almost perfectly which environments are good and which environments are bad. And then we record neurons in their brains.
How do they do that? How do they tell you?
So they tell us that because we don't just deliver water in the good environment. We ask them to lick the lick port for us in order for water to be delivered.
So if in the one environment they're reliably licking and in the other environment they're not, this means the animal understands they're in environment A versus environment B.
Yeah, exactly. There is no punishment for licking in the wrong environment. So in some ways, the animals could just lick everywhere and always get the reward. But it turns out they're conservative with their licks, and they, when they know, right? So then they lick. And if they know they're not going to get water, they're not even going to bother.
Okay. And so what comes next in the story? How do you start to suss out, supervised versus unsupervised types of learning in the
Right.
brain?
So far, this resembles other studies that have been done before, including the neural recording part, which we recorded neurons before this process and we recorded neurons after this process, and we wanted to compare their activities before versus
Mhm.
after.
And you're basically looking at what we would call the visual parts of cortex.
Correct, and multiple different visual areas in cortex. In more or less a big majority of the brain areas that the mouse has dedicated to visual processing, we're recording from all of them at the same time.
Mhm.
And that's where the technology is really, enabling us to be able to do this rather than going and recording from one area at a time over many days, which would be, kind of difficult. We can record literally all of them at the same time, and so have something like a survey of what different brain areas are computing.
Mhm. Mhm. And so what were you guys expect, like, under different hypotheses in terms of how the visual parts of the brain would be learning about this environment? What is sort of the space of possibilities you expected to see here?
Yeah, so I think if we had not been paying attention to what other people were doing, you might think, okay, higher order brain areas should be more plastic, and change their activity as the animals learn a
Mhm.
visual task. Because why would you change, very low level
Mhm.
spatial computations, right? The
Yeah.
very low level units in the brain is for the retina, it's certainly the case. You kind of expect that they kind of deterministically report the same thing, regardless of what's happening. They're
Mhm.
low level units. And so we might have predicted that only higher order brain areas would change their activity. However, um, we already knew from other groups that neurons just about everywhere change their pattern of activity. Not necessarily that they become more active, but they might become more selective between the different environments.
Mhm.
So in the beginning, you might have lots of neurons that respond for the sake of argument. Let's say there's a leafy place in the world and there's a place with circles because those are the stimuli we used. It's
Mhm.
kind of leafy and circly. And there's at the beginning, there might be lots of neurons that respond to both. After learning, a lot of the neurons will be very selective. They'll respond to either one or the other.
Mhm.
So this increase in selectivity, sometimes associated with increases in firing rate, is something that had been reported all across the kind of the visual areas, including at the lowest level of the hierarchy. And so we need to expect that. It looked like that in our data too, as far as increases of discriminate. The neurons are able to discriminate the stimuli better. Um, but we also found one brain area where this increase was highest. And that's kind of where we focused our analysis a little bit more. That was indeed a higher order visual area. Now, I should say that what I've described so far has been kind of how we did actually do the project. We didn't, plan out exactly where we would go from here. We just kind of got to this place. We had all the data. Um, we were trying to think through the data, this kind of massive data set, what could it possibly mean? Until we actually realized there's another crucial experiment we could do um, that would test whether or not these neurons are changing their properties because the mouse is learning a task or because the mouse is just by itself internalizing a model of the world.
Mhm.
And that's what we call unsupervised learning in this case, where we went ahead with a new cohort of animals that were not water restricted. We took advantage of the fact that, like I said, mice like to naturally run. They'll
Mhm.
just run
Mhm.
forward if you leave them there. So they ran, they explored these worlds for also something like 2 weeks,
Mhm.
well, not uninterrupted, an hour a day or two. um,
But there's nothing for them to really learn in the sense of there's no task to do, there's no reward to get. They're behaving, they're moving through the same environment, just like the other mice that wanted to sort of get to the end to get the water, but they had no such thing to learn.
yeah, they had no motivation other their internal motivation of kind of running forward and exploring, and perhaps, paying attention to what's on the walls. And we think they were paying attention to what's on the walls for kind of two different reasons. The first reason is that when we look at their neurons went through exactly the same modifications that the neurons in the task animals went through. So the supervised animals and the unsupervised animals, the neural activity changed in almost the same way. There's a few exceptions. There's one exception to that, but as far as like the system as a whole goes, the tuning properties of the neurons changed almost exactly the same way. And there's a second reason why we think they're internalizing a model of the world, is that if you take these animals that are just experiencing the world by themselves, and then you ask them to do the same task that the supervised animals were doing, now these animals can do that task much faster. In fact, many of them can do that task on the first day, which when it comes to animal training is pretty fast.
How does that compare to the animals that had to learn right away?
Most of the animals that had to learn right away took about a week or so to learn the task. Mm-hmm. Now, but I'm saying that fast, and I think people might have an experience of this because, for example, when I try to train my cat to do things, that can take a very long time and a lot of repetitions of the same thing, or training a dog to do certain things. It takes a while, right? So these mice were actually learning in one day, which for animal training, I'm saying is really fast.
Mm-hmm. Okay. And so the basic idea here is when an animal has an experience with an environment, when it's already familiar with the environment, it's easier for it to learn new things in that environment.
Absolutely. And we think it's because of these neurons that were forming new abstract representations of the world around it.
Mm-hmm. And to what extent were those neurons localized in one place or a few places versus distributed? What can you say about that?
Yeah, so that's interesting because there was one set of brain areas that we call the more medial regions. They overlap to some extent with an area called the retrosplenial cortex. It's a part of the navigation system at least in rodents. It's connected to the hippocampus and other navigation systems. That's the area where we saw entirely new neurons with visual properties develop. So there were very few neurons that even responded to images before. But after this unsupervised learning, we saw like a large population emerge that actually had visual response properties. Now, this was restricted to this particular set of medial regions. But it wasn't the only change that happened in the system. There were also changes that happened in the primary visual cortex, in other high-order visual areas. It's just that these areas already had neurons tuned to stimuli before. They were selective to stimuli before, especially primary visual cortex responds strongly to visual stimuli. It's just that the patterns of responses, the tuning changed. And the tuning changed in kind of a gradual way towards a place where when an animal was experiencing the same kind of repetitive corridor for many, many days, it would start to essentially develop an entirely new dimension of neural space just for that corridor. So like the same way we're talking before that, neural computations are extremely high dimensional. It turns out that over time, things that repeat a lot, things that we might form into visual memories, kind of start to stick out from the cloud and develop their own dimension that's orthogonal to all the other dimensions that existed there before. And so this process is kind of, it's more one of changing the angle of a population vector rather than introducing a whole new set of neurons like what we saw in the medial areas.
And what might the behavioral advantages be of that type of operation. It's intuitively, you might think, okay, so the brain is sort of making it easier to pick out this thing that you're very familiar with, and therefore probably have to interact with more or something like that.
Yeah. I think the gradual nature of the process, where it actually took kind of a couple of weeks for this dimension to kind of really stick out and be completely separated, it's the gradual nature of the process that gives us the insight here. Because in the beginning, let's say animals run through this kind of leafy corridors, there's leaves on the walls. If we put them into another corridor that also has leaves, but kind of in different places, different, doesn't, it looks the same kind of abstractly, but it's a different location, let's say in this forest. Then they still respond in that environment as if they think that there should be reward in there. Like they respond in a similar way. And that we think is arising from the similarity of visual representations, right? So as you're forming a memory of a place visually, you might be forming, some abstract, leafy places are all good. They all give me rewards. So whenever I see a leafy place I should lick, I'm going to get reward. But then with time, as you start seeing the same place so many times, it gets kind of seared into your brain, right?
Mm-hmm.
I think it's an experience a lot of us have when we recognize something that we've seen a lot, just kind of see a pattern and we know we've seen this before, right?
Mm-hmm.
That is called visual recognition memory.
Yes.
As visual recognition memory develops, it develops for very specific exemplars. It develops for specific instances of the world, not for like the general concept, not just for leafy places in general that are good. This particular leafy place where, there's a tree to the left and a tree to the right and some, bush in front, this place, this is what's good. And that requires a very specific memory for that particular pattern. And we think this dimension kind of sticking out of the cloud is an instantiation of that memory being formed and being very specific to that place.
Yeah, it's a way to make a place stick out because it's important because, you get water there or something good or something like that.
Yeah, and you've seen it so many times that you can recognize like very small details and know if a small detail has changed.
Yeah, and presumably the memory of that location is much higher resolution than other locations.
Yep. Exactly. It's a high resolution, version of it inside of your internal model.
And when we talked about supervised versus unsupervised learning, I was starting to think like, okay, obviously we can discriminate between supervised and unsupervised when you guys create a whole situation where you introduce the labels or the conditions for the reward. Like it is supervised in that sense. But actually, when you step back and think about it, especially when you think about it for simpler and simpler organisms, in what sense do they ever get like something supervised? Like almost everything in their life is going to be a form of unsupervised learning. Is that reasonable to think about?
It gets almost everything in their life will be a form.
Yeah. In what sense is there supervised learning for like a fruit fly in the real world?
So I think that's where we bring in something, called reinforcement learning, right? Which is, it's often being distinguished from these two. Reinforcement learning is more about, getting reward, getting good outcomes for actions that you take. It's kind of more inherently of a kind of a low dimensional computation because you only get so few rewards in the lifetime. But then if you have a good algorithm, you can kind of figure out what led to that good reward. What sequence of actions led to that good reward. How does that relate to the sensory environment?
And I suppose in the absence of having an external agent like an experimenter or like your mother or something, if I think about an animal who's taught by observing its mother, in the absence of having those kinds of labels, I guess in a sense, if certain sort of rewards are baked into the brain just intrinsically by evolution, those can be thought of as just preordained labels. And so they're supervising the learning in that sense.
Absolutely. And every organism that we've looked at has had a big part of its brain dedicated to monitoring for those rewards and to kind of associating good things that animals do to those rewards.
Yeah, yeah.
Part of it is the kind of the dopamine system that people talk about a lot. It's a very kind of widespread signal that gets distributed throughout the brain. It's, if you want, it's the it's the learning signal essentially
Yeah.
for a lot of these, even for the higher dimensional computations.
Yeah. And I mean, this is where you would talk about, so, dopamine, it's associated with motivation and the, obtaining rewards. But this is where oftentimes this is like the first sort of computation you learn about is the reward prediction error, which is, I mean, if you want to describe it for people, it's actually a powerful but remarkably simple phenomenon. Can you talk about that a little bit?
Yes. So it also has to do with the brain making predictions about the world.
Mm.
So let's say you have this complicated machinery we've been talking about so far that makes a model of the world, and it's able to predict based on the model of the world that certain things are good and certain things are bad. And then you get into a situation, you predict, okay, something good is about to happen because, I don't know, I walked into the elevator, I pressed the bottom for my floor, and I expect it will go to my floor. I don't know, something like that. But then all of a sudden, something unexpected happens, and the elevator just doesn't move, let's say.
Mm-hmm.
Then we would say, if we were to record from certain neurons in your brain that are dopamine neurons, we would possibly see a spike, a reward prediction error, a spike of neural activity associated to that moment when we realized that something that was supposed to happen didn't happen.
Mm-hmm.
Now, the way we study these in a lot of animals is related to rewards because that leads to some of the biggest spikes. If an animal expects a reward will happen, like Pavlov's dog, when a bell rings, and the reward doesn't happen, that's when we see some of the biggest such spikes that are reward prediction errors.
Mm-hmm.
And it is thought that this signal is especially good for learning because if your model was so sure that something good was going to happen, but it didn't, it means your model was wrong. Your internal model was wrong. You need to adjust it. To adjust it, you need a signal. The signal is the reward prediction error that then gets distributed kind of across brain areas, and through magic we don't know, is able to change that internal model to say, sometimes there might not be a reward. Get used to it. That's fine. It's normal. It's the new normal.
There's some kind of comparison going on between, you've got a model because you're like, I have an expectation of what things should look like in the world, and then they don't look that way. So the expectation is violated. It's some kind of like subtraction like operation.
Yeah, and that happens in advance, right? The model predicting reward is something that is a recognition of a situation where if you just do this or if you just do that, you might be able to get a candy bar or the machine, right?
Mm-hmm.
And then it's when that has already been distilled into the expectation, something good is going to happen. If it doesn't, candy bar does not come out. We have a reward prediction error. We have to go back in time, figure out what parts of the prediction were wrong.
Mm-hmm.
Was it, this is not actually maybe a machine dispensing candy bars, or maybe I didn't put in the right amount of money, or maybe it's out of candy bars and for this particular thing I usually get. So we have to still figure out which of those parts of the model was wrong and then correct accordingly. That's where it gets complicated, right? This initial part of a reward prediction error distributed to different brain areas is by no means a simple thing, but then actually associating it with the internal model is where it can get really hairy.
Mm-hmm. In the little bit of time that we have remaining, I just want to ask you some general questions to get your perspective as a neuroscientist and as a computational guy on things like the large language models and the AI systems we build in the digital artifacts that humans have invented. High level, just your general take on this stuff. When you look at the large language models that are out there, people are using ChatGPT every day and all of the other LLMs that are out there. They can obviously do very impressive things, but to what extent are they how would I even say it? To what extent are they actually reasoning, or do you think that they're implementing the types of algorithms that biological systems like brains are doing? Can you just kind of compare and contrast, or maybe tell us if you think something, is there something sort of fundamental that we think brains are doing that the LLMs are not, or vice versa?
Yeah, I mean that there's all sorts of things to talk about there. Um, I can give you more of my perspective on it rather than reflecting a more general perspective. I think the
Mm-hmm.
more general perspective in neuroscience, right, is that um, for better or for worse, right, that there's something more special about how brain computations work that we haven't really captured the magic of reasoning into the artificial machines yet, right? And then the critical part of that becomes the yet, because some people think you can scale the current systems to a point where they do become looking like more like reasoning, even if currently they don't do that. Um, Now, my perspective on it is that humans and the kind of reasoning we have has been arrived at through evolution to some particular idiosyncratic version of reasoning that we have, where our brains are it I don't think our brains are like doing it in a simple like correct way. I think they're doing it in this very convoluted way, um, where it just kind of happened through evolution to get us to this point. I think it'll be very difficult to replicate that in a
Mm-hmm.
machine
Mm-hmm.
because it's reverse engineering our brains, which is what we do in neuroscience, but very convoluted, very like strongly connected network. It's very hard to reverse engineer. But that doesn't necessarily mean that whatever version of reasoning an artificial system gets to isn't reasoning. It
Mm-hmm.
just might be a different version of reasoning. It might
Right.
not be what we do. But as far as like outcomes go, we can still argue whether the outcomes are, the responses that LLMs give us are good or bad. And
Mm-hmm.
that's really most of what we argue about, right? Is the response is good or bad. Now, I think one part of that, though, that I think ties more closely to my research that I think is interesting to mention is that the way LLMs are trained is technically something people might call self-supervised learning, um, in the sense that LLMs are not, well, they're trained in multiple stages. The main stage, the more like self-supervised stage, is the one that takes the longest. And it's technically not done with supervised data. LLMs do not have, well, they are fine-tuned on relatively big data of questions and answers, but that's not all they've been trained on.
Mm.
That's just a fine-tuning step. It's a relatively small part of how these LLMs are trained because they're initially just trained to predict the next word in every sentence, right? And that is what we will call a self-supervised training because we can just give it books and websites and emails and everything, and we don't need to label it
Yeah.
specifically to say, this is a particular form of communication, or uh, it can just learn by predicting the next token, the next word in the sentence.
Yeah.
And the reason why that's neither supervised nor unsupervised is we don't give it specific labels, but it makes its own labels in a sense.
Mm-hmm.
It's still using the same kind of supervised learning algorithms, the deep neural networks that take an input and produce an output, but it's making its own labels of what is it going to predict. It's making its own task of predicting this next word. And that's where I think a lot of the kind of frustration with LLMs comes in because um, in principle, predicting the next word, if you can do it really, really well, can be a mechanism to generate entirely long sentences. Instead of predicting the word, you produce the word and you produce the next word and you produce the next word. That's how LLMs work. But then as far as like researchers, especially those of us thinking about the brain go, that is certainly not how, humans are trained to reason by predicting the next word, right? We reason across much bigger future timescales. Also, we have pretty long timescales into the past, but LLMs have that as well. What LLMs do not have easily right now is the ability to kind of do a lot of kind of forward modeling and like pick the best version out of those. And the best specialized models that we hear about that do some of the most impressive tasks, like solving very complicated math problems, like international math problems and things of that sort, those are the ones that do have some of this forward reasoning. And yeah, I mean, I think that's kind of my perspective on it. Just it's another computational system. It doesn't have to be the same as what humans do. It can be studied for kind of what it is. And it will be very interesting to see just how, far these kinds of systems can get to. For us neuroscientists, I think we mostly think of them as like another interesting system to study.
Yeah. It's another system, just like you're studying multiple systems in biology. You've got different species, different strains. Obviously, in the realm of biology, you've got, evolution by common descent. So they're all related to each other. This is almost just like an alien branch of we might think of as a phylogeny. And so therefore, it might just have more distinctive properties. It might work in different ways because it has like this independent origin.
Yeah, exactly. And that kind of contrast can often lead to kind of realizations about how the systems work, what's necessary for certain properties and what's not.
Given how much of the brain is about the body, in a sense, right? You've got to move this body through space to survive and propagate. Much of the brain is about learning things about the environment insofar as they can inform patterns of muscular contraction that the neurons are generating. The LLMs don't have that. They don't have bodies and muscles like that. So how does the sort of embodiment or disembodiment play into this? Do you think that's an important thing in terms of what these systems will and will not be able to do?
I think my perspective on this is that the embodiment problem is certainly a difficult one, and one where humans and animals, that's where we still are much better than anything else, right? So far, we've been able to, even a few years ago, right, we would say that it's our cognitive abilities that like set us apart. But now we have LLMs that can mimic some of those things that we didn't even think was possible before. Our motor abilities are still, far from being reached by robots or any kind of, artificial systems. But I think a lot of that has to do also with our biology, with how efficiently our cells are working. Some of those principles you started with, right? The efficiency of them, they tend to apply quite a bit more to other parts of our body that have been kind of optimized through even much longer evolution before that. And so while I do think that this is a hard problem, that's not, it needs a lot of work to be solved on the robot side, making robots with, LLMs that are sufficiently embodied. I personally do not think that is what sets us as humans apart. I think it is those internal models and our ability to reason. We can do very sophisticated things without moving a finger, just by thinking. People can, write entire novels about continents they haven't been to just from their, Paris apartment or something like that. And I think, a lot of those kinds of properties are things where we have built our societies around, and we have built, it's why the internet is such a drain on our attention, is because it connects us to a lot of knowledge and a lot of new things from all over the world. I don't think it is our, muscular systems that are going to be where, LLMs are suddenly going to become conscious because they are embodied. No, I don't think so.
Interesting. Well, I think that's a great place to leave it. Your work is fascinating, Marius. I want to thank you for your time. I encourage people to go check out your work and work sort of in the realm that you play in if they're interested in learning in biological systems, but also how some of these computational principles apply and some of the crosstalk between the ML world and the biology world. Is there anything you want to leave people with? Any final thoughts or things you want to reiterate from our discussion today?
Well, I want to thank you for inviting me again. It's great to be on your podcast. Um, I think the only thing I would iterate is that not the specifics of what we talked about, but rather I think the fascination around studying this system is real. It can be very exciting, especially if you're a student, if you're someone who's like me, 15, 20 years ago, looking for, some kind of interesting field, right? Because I had these mathematical skills that I felt like it wasn't that useful. Solving math problems, the kinds of pure theory math problems that professors of mathematics solve isn't directly impactful into the world right now. But you can take a lot of those same kinds of skills and you can apply them to these fascinating systems and study them. Whether you're trying to understand how brains work or how machines work, it's these complex systems that kind of have fascinating us for so long that I think are really worth getting into, because they can keep you motivated for very long periods of time, for many years, and probably for entire lifetimes and careers.
To read like I do, then you should check out the Readwise app. It's totally free to download, and if you go through the link in the episode description or on my support page, you can get a free 60-day trial for zero cost. Readwise is a really cool app that allows you to import digital highlights from over 30 different platforms such as Kindle, iBooks, PDFs, and more. It makes it really easy for you to review and find and share excerpts and quotes from whatever it is that you're reading, whether it's PDFs of scientific papers or books that you're reading on your Kindle, and you can even save and use highlights from physical books. I read a lot of physical books before bed, for example, or when I'm traveling, and the Readwise app actually integrates with the camera on your phone so that you can take a photo of a page that you're reading, highlight a specific chunk of text from that page, and export it to the app so that you can reference it or find it or share it later. They even allow you to export really cool looking graphics of some of the highlights that you have in the books or the papers you're reading so that you can share them in an engaging way. So check out the Readwise app today if you are a reader and you like to reference and organize what you read. Go through the link in the episode description or on my support page or in my free weekly newsletter at mindandmatter.substack.com and try the Readwise app 60 days free today.